in a short video lecture from microbiology with Chagnik, where we are going to revise our concept regarding RT-PCR. So RT-PCR, the full form of RT-PCR is a reverse transcription PCR. With the help of reverse transcription PCR from a cellular or tissue RNA, we can have the cDNA, and we can also amplify and isolate or identify the known sequence. Okay, so here the PCR reaction will be preceded by a reverse reaction that will be catalyzed by reverse transcriptase. Okay, this reverse transcriptase will convert the mRNA into what? Into cDNA molecule, and later the cDNA molecule, as normal PCR procedure, with the help of normal PCR procedure, can be amplified. Then with now by simply using this amplified product, we can isolate or identify our desired sequences, right? So that is the idea of reverse transcription PCR. But here remember, instead of using reverse transcriptase, we are using those kind of DNA polymerase that can act as a polymerase that can use RNA template as a substrate. Okay, and also that can act as a DNA dependent DNA polymerase. That means here we are targeting to complete the two step by using one enzyme. So an example of that kind of enzyme is the recombinant form of TH polymerase, that is from Thermus thermophilus, which can catalyze high temperature reverse transcription from mRNA to cDNA molecule in the presence of MnCl2. That means it has the ability to perform both intrinsic reverse transcription as well as thermostable DNA dependent DNA polymerase activity. So that is the idea. So remember the one thing, that reverse transcription procedure is preceded, it is as same as the normal PCR procedure, just only it is preceded by a reaction that is catalyzed by reverse transcriptase, and this reverse transcriptase converts the mRNA from the cellular or tissue RNA into cDNA molecule. Now this cDNA molecule will be amplified with the help of normal PCR procedure. Okay, and now this reverse transcription, instead of using reverse transcription transcriptase, we are using a type of enzyme that can play the role of both the DNA dependent DNA polymerase, as well as it can act as a enzyme that will use RNA template as a substrate. So an example of this kind of DNA polymerase is Thermus thermophilus, that can catalyze reverse transcription at a high temperature in presence of MnCl2, and also perform the thermostable DNA dependent DNA polymerase activities. Okay, so that is all about reverse transcription procedure. I hope that this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. And I will again come back with the real-time PCR video. Okay, so goodbye.